I thought I'd do is tell you a little bit of where we are with biomarkers for hepatocellular cancer, and then our work, and hopefully bring you in, and then perhaps a proof of concept clinical trial that might entice you. And Ben Dar is someone I've been talking to at length, and hopefully he will lead something like that. The biomarkers first. So as you know, this is a big problem for us, right? For hepatocellular cancer, ultrasonography, which we use, has limited sensitivity. And now that a third of our patients have obesity and NASH, this actually is even reduced. And moreover, most people just don't go for their sonography and then raise alpha fetoprotein. This isn't high in about up to a third of hepatocellular cancers. And apart from longitudinal studies, elevated AFP is limited as an independent prognostic biomarkers for hepatocellular cancer detection. So there are quite a few biomarkers in phase 3-4, such as this highly reactive alpha fetoprotein L3 and des-gamma carboxyprothrombin and midkine and Dickoff and glypican-3 and osteopontin and acetyl nitrite. And there are microRNAs and DNA methylation markers and a proteomic analysis of tissue interstitial fluid and extracellular vesicles. However, while these are in phase 3-4 trials, there are many problems. And these are, these cancers are heterogeneous. There are multiple etiologies, divergent pathophysiology. And how could you start beginning looking at this? So many years ago, we said, well, perhaps you could really start mirroring in the blood what was going on in the liver. And our work in the TGF-beta pathway led us to thinking that. So for instance, if you disrupt the TGF-beta pathway, TGF-beta is very pro-metastatic in humans and can turn on a fire, but the mouse models show the opposite. And perhaps we could start thinking about this and looking at alterations. And then how are we going to do this? And how are you going to do this when there are many hospitals are in the middle of nowhere and have no access to a high-powered analysis? And if you had a choice, if you're looking at proteomes, how are you going to take this further? I mean, is DNA the place to go to? RNA, mutations? How are we going to really begin looking at biomarkers? And this has been a long thinking hat, but ultimately the worker horses in our bodies are the proteins. And I think Ray Shinazi had a meeting that he convened many years ago, and he asked everyone, is DNA the way to go? RNA the way to go? Epigenetics? And a few people said proteins. And the reason why we've never focused on proteins is because we've had limited access and antibodies are very expensive. So I'm going to take you on to a little journey that's taken us in the protein direction. So we formed a consortium with Maryland. At that time, Kirti Shetty was at Hopkins, University of Hawaii, because they had a large patient population, which was diverse, Herbert Yu and Linda Wong, and MD Anderson, where I had been working beforehand, to look at real functional biomarkers for early liver detection based on what we found. And we also harnessed Xinwei Wang to look at these, and he's intramural at NCI. So the whole goal is to identify those patients at risk and then follow them up for hepatocellular cancer and determine their molecular characteristics, and then analyze the risk. And the high-risk patients, targeted treatment, low, less invasive treatment. And so, you've got this, what has happened for many cancers, particularly lung, prevention, early detection, prognostic assays, and then a substantial cost saving. But it requires all of these: biomarker panels, blood-based biomarker panels, validation, large-scale analysis, and then, of course, clinical trials and clear certification. It's a long road. And for liver, we are far behind the rest of the cancers. And of all the cancers, the liquids have shot up to the forefront, and of the solids, hepatocellular cancer remains in the bottom pile. So how do we get ourselves out of it? And so we did the hard approach. We really went to discovery. We looked at what was going on in the liver. I don't know how many of you are familiar with the Cancer Genome Atlas, but the TCGA data analysis has been very instrumental for us. I think in 2008 or 9, from that point to 2016, NCI literally poured money into high fidelity 60 to 80x sequencing of a vast number of cancers, mostly obtained in the United States, but for hepatocellular cancers, many of them came from Africa and other parts of the world. And these samples were annotated, analyzed, and then published. And these are a series of Cell publications, which we played a role, and we described the TGF-beta pathway in those. And here's the powerful part of this. It's very expensive, but it's an objective analysis. And it's been shown that if you write an NIH grant and you have TCGA data, which is really objective, supporting your studies, you say there's this X protein, but osteopontin, for instance, but it's so instrumental in hepatocellular cancer because of the TCGA data showing mutations or epigenetic regulation, your chance of getting that NIH grant or securing it are threefold higher. You really bump up the likelihood of success. This is such a powerful database. Of course, there are many other databases, and CBioportal, which you can access on your cell phone, really collects all that, and that's from Memorial Sloan Kettering, and they've done that. So we looked at these TGF-beta markers, and then we looked at a number of others, which I'll tell you a little bit about: epicam, osteopontin, polar-like kinase, vimentin, DNA repair pathway. And then our goal was to really look at tissue and see if the tissue and what was going on in the liver reflected anything that could tell us about risk for hepatocellular cancer. And once we had done this for tissue, we would look at blood and look at serum biomarkers for all of these, including circulating tumor cells. Okay, so we started off with 10 functional, what we call functional biological biomarkers. When you disrupt any of these molecules, you get a cancer activated or cancer suppressed, or they seem to be instrumental. The TGF-beta type 2 and the type 1 receptors have been shown. If you take mouse models, these mice get cancers. If you lose these, they're by TCGA data are very supportive of these being instrumental in hepatocellular cancer. Arspondin-3 is an interesting protein. It's Wnt related, and it's also found in like osteopontin in the stroma, and so it's pivotal in hepatocellular cancer. Spectrin, which we cloned, is a TGF-beta adapter, and it's lost in the hepatocellular cancers in a heterozygous situation. In the homozygous situation, it's another story, and it's really interesting. Serum intertrypsin alpha-1 inhibitor, which we had also cloned, but a group in France that's shown to be very instrumental in NASH. HMGA2, high mobility group A2, this AT hook proteins, these proteins have been shown to be important in the stroma. Epicam, of course, you're all familiar with its stem cell role, and a beautiful cancer cell publication from Xinwei Wang, as in polo-like kinase. So epicam is expressed in stem cells, and polo-like kinase is instrumental in a C1 group of Asian liver fluke infected hepatitis B that progressed to cancer. Sirtuin-6 knockouts get hepatocellular cancer, and Fanconi D2, we had identified through TGF-beta signaling a DNA repair pathway being promising. So we really looked at these 10 functional biomarkers. As you can see, they're widely different, but they seem to target a progenitor cell in the liver, and perhaps their loss or their alteration leads to hepatocellular cancer in mouse and human. Okay, so I'm just going to really fast forward. And what we did was each institution, George Washington, then we classify them as GW1, GW2, GW3. This is the George Washington University UO1 consortium. And so one of them is George Washington University, the second is University of Hawaii, and the third is University of Maryland. 3 independent cohorts. Now, what we did was we sent out antibody to each of these, and they examined their hepatocellular cancers in cirrhotic patients for all those 10 antibodies, the type 1 receptor and the type 2 receptor. We then had 2 pathologists analyze these samples, and then we crossed over. We shared samples across, we shared tissue, and the standard operating protocols. We developed one of those, and MTAs, and then we crossed over and determined whether the same set of patient samples could be reproduced in all 3 institutions. Okay, so this is immunohistochemical labeling. On the top left is a TGF-beta type 1 receptor. If you look at hepatocellular cancer, this receptor is absent. It should be present in the membrane and cytoplasm, but not in the nucleus of normal cells or cirrhotic cells. In cirrhosis, this protein goes up. It's as though the cell's trying to rev up and make more, but it's not in the nucleus, suggesting that the type 1 receptor is functional, but not effective. And subtly in hepatocellular cancer, it's lost. It's not lost in all, but in a large number. The type 2 receptor seems far more dramatic. It's really lost, seen at the bottom left. It's lost in this high power, and in cirrhosis, it's raised. Okay, so on the right, you see these samples. These are 20 samples adjacent to non-tumor, hepatocellular cancers, and cirrhosis. And as you can see, the hepatocellular cancers for the type 1 receptor, to an extent, seem decreased, but not significantly. In one of the institutions, it's markedly altered. And it may be that this is Hawaii because they have a very different population, an Asian population. You take the type 2 receptor, this was decreased in hepatocellular cancers across the board, and this could be reproduced across all 3 institutions. But this is manual. You take these cirrhotic patients, and then you label them for antibodies, and then you look, and this is a long, laborious, expensive course. And could we hasten this? Supposing you are on Kauai, in these remote Hawaiian islands, and you have a pathologist you have a hepatocellular cancer, you're wondering, you have a cirrhotic population, you're wondering, are these going to develop HCC or not? This is not a likely test that you're going to be doing, right? It just isn't. We know the type 2 receptor is reproducible. So what we did was first we did a manual analysis across for the type 1 receptor, the type 2 receptor, and many of the other samples, as I'll show you. Richard Amdur, we have a strong biostats and bioinformatics program, and largely at GW. They looked at the sample size, and then they did a power analysis, and then they compared the staining, and they used a multivariable logistical regression. Now, it's called a Kowalski test. It's not your normal student t-test, so that it really addresses these outliers and all the noise. And this was very rigorous. And it really turned out to be positive for the type 2 receptor. But along the way, I'd just like to show you labeling for epicam, the stem cell protein. This is adjacent to hepatocellular cancer, cirrhotic liver. Epicam is really strongly positive in these unusual cells. They do not look hepatocytic, but they're really in the portal tracts. And hepatocellular cancer, its labeling is really altered, and it's diffused throughout the cancer. Polo-like kinase seems to go up in hepatocellular cancer compared to cirrhosis. Now, this labeling to us was all over the place, and we didn't really know what to make of it. HMGA2 could be promising. It's raised in hepatocellular cancer compared to normal cirrhosis. And R-spondin is the reverse. R-spondin, of course, is a stromal protein, but it's really, really raised in hepatocellular. in adjacent cirrhosis and then gone in hepatocellular cancer, it seems. So a lot more work. SIRTUIN-6 all over the place, ITH4 all over the place, and spectrin, perhaps to an extent, you see this strong nuclear localization in hepatocellular cancer, which we didn't know what to make of, and FANCD2 all over the place. So you can be wildly unsuccessful, as we were, with many of these biomarkers. It's sobering. It was a lot of work, and it took the first 2 years of this NCI-funded UO1 to look at these 10 biomarkers in this rigorous manner. And so going back, what are we going to do? We've got this population in Hawaii, in Kauai, and this remote part with cirrhosis, and you want to know whether they're high risk. And we thought, well, maybe we could look at artificial intelligence. And I don't know, are any of you familiar with this company called Aitoria? It's Finnish, and they work with a huge European NASH cohort. And there's a woman who leads that. She's out of Helsinki. And so Aitoria stands for Artificial Intelligence for Intelligence Analysis. And it's this company that really standardizes it. What it does is you take these samples, seen in the bottom left, you scan them, slides, you label the slides, and you scan them. And then you use a machine learning tool, and you can send this, it's cloud-based, and you can send this data goes to Helsinki, and they have a team, they have an image-based analysis, and it's machine learning tool. And then this is validated by pathologists, researchers, and a number of people. And we entered into a collaboration with Aitoria 2 years ago, just before COVID, and to look at our samples and to see if we could now do something remotely. And, you know, Nazia, if you all the way out there you had some slides that you'd labeled, you wanted a quick analysis within 24 hours, this could be an approach. And they have an office in Massachusetts. So it enabled, we thought, a standardized, accelerated, and inexpensive preclinical investigational analysis. And could it be accurate and reproducible and use a cloud-based platform? So this is what they do. Here's a sample seen on the left. You have immunohistochemical labeling. And this is for one of the receptors, the type 2 receptor, I believe. And you have a very cirrhotic patient with cancer, right? And what you have is adjacent tissue, cirrhosis, hepatocellular cancer, negative, strong and weak signaling. And this labeling, this image analysis really quantitates all of this and quantifies these and on a 3-tier system. And then they then give you an 8 score. And here you can find to the right that for the type 2 receptor in the first 20 and then in ensuing samples, the type 2 receptor is considerably decreased in hepatocellular cancer using this platform, showing that it can be successful. Now, in the process of really asking them to validate this in the larger cohort, but that's a long story in itself. But we thought, okay, we're successful with immunohistochemical labeling, but this is still very expensive. And could we go to blood? Could you go to the middle of nowhere and take a little bit of blood and send it off for analysis? Well, where are we? So again, pre-COVID, we partnered with a company called SomaLogic. I don't know if any of you are familiar with SomaLogic, but it was spurred off by Novartis, and then it's a company that is in the heart space a lot. They have CLIA certified markers for coronary artery disease. But what they do is they look at proteins in the blood, and they only take 130 microliters of either serum or plasma. And in the process of developing a strong signature for NASH, and I don't know if any of you are involved in that huge NASH study, but they have thousands of NASH patients, and they've identified in 130 microliters of serum that you can have a signature that can tell you that you have severe NASH. And that is looking at how many proteins. Initially, they started with 5,000 proteins. Now they're at 7,000 proteins. These are aptamers. And so they don't really, a multiplex antibody screen is very, very expensive. But this is much cheaper, 7,000 proteins in one test. Now they sell this for quite a lot. They sell it for $500 a shot. And if you're NCI funded, it's cheaper, but it's still not cheap. But Medicare seems to pay for the heart tests, and it's CLIA certified. So we use the first 170 samples. We used high-risk cirrhosis patients, and then we used hepatocellular cancer patients as controls. Let me give you patients with cirrhosis and look at the TGF-beta family. So we used our TCGA family of TGF-beta regulated genes, seen on the vertical axis, and seen here from FOXM1 all the way to inhibin A. And as you can see, all our favorite proteins, SMAD3, SMAD2 are in here, collagens, which are regulated by TGF-beta, and so on. So this is through the literature search and through cohorts. These are really considered bona fide TGF-beta pathways. There are about 283 in the Soma logic group that we can look at. And as you can see, if you look at cirrhosis, there's a definite signature. These, in the top panel, a number of proteins from FOXM1 to RAN to SMAD3 are switched off in cirrhotic patients. Whereas MMP9 collagens, MMPs are really activated in cirrhosis, which makes sense, right? Because these are molecules involved in inflammation. And then in cancer, these molecules, of course, switched off. And then you have the pro-cancer agents being switched on. And surprisingly, SMAD3 and SMAD2 are harnessed, BMP4, these are DICOF, these are all harnessed to turn on cancer. So in our cirrhotic group, we had 14 patients, to our surprise, that looked just like the hepatocellular cancer signature. Now, these 14 patients by sonography and by alpha-fetoprotein, at this point when this blood was taken, did not have cancer and had no hepatocellular cancer. Now, we think that this could be a hepatocellular cancer signature in cirrhotic patients that might signify an early cancer. And so our next study, which we are part of this huge UO1 consortium, is to look at LR3, LR4 lesions, these 1-3 centimeter lesions in the liver identified by MRI. And there's a 2-year follow-up to see which ones progress to hepatocellular cancer. And ideally, we'd like to see if this signature can be recapitulated in those patients, or whether many of those which do not become LR5 lesions, LIRAD5 lesions, 4-5 lesions and progress are really the cirrhotics, and others are really this. So it'd be really interesting to see through this prospective study that we've, the part of the UO1 consortium is with Michigan, UT Southwestern, UCLA, and Baylor, and then our consortium together. So it's a large consortium of which I really hope that the panel of people I'm talking to today will really be much larger, and this work, we could take this to another level, because NCI is definitely interested in funding this as a clinical trial independently of that UO1. So I hope I can get you all interested and stimulated to join. And here are these signatures, biomarkers, that can potentially distinguish cirrhosis for hepatocellular cancer. And we think it's just this small group of patients. And then we looked at HMGA2 and ITIH4 and polo-like kinase and R-spondin3 and the type 2 receptor. And to an extent, there is a difference, but that still has to be tested and looked at further. Soma has published a lot of its work in Nature Medicine and gone to CLIA certification for its work on coronary artery disease. And now I think they have a paper in review on the NASH in Nature Medicine, but I'm not quite certain about that. Now, what about previously tested biomarkers? I told you about a number of them, which are in clinical trials. Glypican-3, seen over here on the left panel, which is in hepatocellular cancer raised, but it's also raised in cirrhosis. And I think of all of these, FGF19, FOXM1, and all of these, I think we need larger numbers of patients. These are 168 patients. We are now progressing to the next set. And from Northwell, we hope to have 400 in the next year with far more stringent controls, such as breast cancer and gliomas. And then the fibrosis-related genes. And as you can see, the collagens are highly activated in cirrhosis, and they're largely switched off in cancer, which is food for thought. So something other than TGF-beta to look at. Okay, so these 108 biomarkers for 168 patients. How did we look at this? So Richard Amdur, who's our biostatistician, examined distributions for skewness and outliers, and Raja Mazumdar and Keith Crandall took it also to a machine learning tool. And we discovered many things. We discovered that there's a false discovery rate. It creates quintiles. We used a Kruskal-Wallis test. They used that, and they looked at cut points and then tried to improve the model and carry out a machine learning tool. And out of all of this, they found that what we had identified through the manual analysis was indeed robust for the type 2 receptor, and that this signature that we'd identified through SomaLogic was really strongly valuable. So that was really interesting, when we'd really like to take this out to the next step. So in this first part, I think what I've shown you is that there's a reproducible biological, what happens in the tissue can be reflected in blood-based analysis. And we can do this across multiple sites in a rapid, objective manner by tissue, which is slow through Aitoria with a type 2 receptor, just one receptor being promising, functional predictive hepatocellular cancer, but far more exciting is this objective panel with a very low amount of serum or plasma. We are in serum because the NASH patients and the NASH cohort that they have is all in serum, and with only 130 microliters, which we'd like to take to the next step. So where is the future? With all of you, we'd love to take this to really testing large numbers and then community-based imaging to correlate whether these cancers really happen. And we'd really like to validate these blood-based biomarkers and scale up to a clinical trial. And I think we are really there. Bob Miller, the Dean for Research at GW, actually has been very proactive about this. And he's taken a number of therapeutics from his time at Case Western to clinical trial successfully. So I think this is something we are here for these biomarkers at the bottom, the future. We are now here. And I would now ask this group to bring this forward, and whether we could do this together. So let me take you to the next part, which is, I think, for us, very exciting as well. What about the pipeline? How could we look at obesity, NASH, and hepatocellular cancer? Clearly, this is a global epidemic. And we have found a very surprising phenotype. We have these mice, which we call ASCO mice, that develop metabolic syndrome. They're obese, have high triglycerides, low HDL, high glucose. And as you know, a quarter of the world's population can be obese. And a number of these people have metabolic syndrome. And of course, 20%, a quarter of these will develop NAFLD, and then 20% NASH, and then a smaller group, hepatocellular cancer. And this is an $84 billion market with 4 therapeutics in phase 3s, but no single agent, right? What is the pathophysiology? The whole idea of working in mice is that you can go back and test systems. And so we know from all our work in cell lines and mouse studies that in obesity in the liver, which is an awful amount of stress, generates endoplasmic reticulum stress and activates TNF-alpha and caspases. And instead of cells dying, you do get some cells dying,
But what happens is the hepatocyte then becomes very aggressive because SRABP1, which through which Brown and Goldstein obtained their Nobel for discovering this, is that master regulator, which is switched on, and lipid formation is activated in the liver. So liver reacts to injury with two things. The right activates TGF-beta with fibrosis, and the left, you get lipids and lipogenesis and steatosis and steatohepatitis and NASH. And together with fibrosis, you get cirrhosis and cancer. So we are on the right-hand panel, but what about the left-hand panel? And what happens? So about 6 years ago, we carried out, maybe even longer, 8 years ago, we carried out loss of function studies in our TGF-beta knockouts and found that aldehyde dehydrogenase 2 was markedly raised. Now, ALDH2 is really exciting because it's a clearinghouse for alcohol. It's a clearinghouse for a lot of things. It's highly expressed in the liver and is involved in DNA repair. So it's an NADP-dependent mitochondrial enzyme. What's even more interesting is 500 million people are affected with deficiency of ALDH2. And these are Asian flush syndrome. There's a cohort in Sweden. They have this, there's a functional deficiency of ALDH2 with this isoform ALDH22 in these people. And of course, it's associated with a number of things from Asian flush syndrome, cardiovascular disease, Alzheimer's, osteoporosis, and many cancers, esophageal, liver, and head and neck. So we found aldehyde dehydrogenase 2 was raised, and we thought, well, in our system, why don't we intercross the ALDH2 knockouts with the spectrin and the SMAD3 knockouts, and we'll see an alteration in hepatocellular cancer. And we were really interested in liver cancer. But to our surprise, Shu, who's an assistant professor with me, intercrossed these mice, and she came up to me and she said, you know, this is really weird, because these knockouts seen on the far right, as you can see, they're white. They're very, very, very fat, and they're very, very obese. They have this truncal obesity seen here on the bottom left, compared to wild type normal mice. And this is on a normal diet that they're very obese. And you know, you and I are always used to telling people, you can't get obese on a normal diet. And people will say, I eat nothing, and I'm still getting fat. And perhaps this is the reason for it. Anyhow, this really shocked us. And so we explored this further. Of course, these mice get metabolic syndrome. We never tested blood pressures. I don't know how to test blood pressures in mice, but they have everything else. The waist circumference, triglycerides, low HDL, high glucose. And then we found that the liver-specific knockout, when you get rid of this spectrum completely, it disappears. So we thought, well, how about proof of principle? How about an siRNA targeting the spectrum? Now, this was before COVID. And we found if you have this control mouse, this ASCO mouse on the left, which is really fat and has, this is fat on the small panel on the left, and you give the siRNA, that fat decreases and it reverses this obesity. And so here, focus on the right-hand panel of immunohistochemical labeling. As you can see, there's steatosis in these normal diet fat mice. And with the siRNA, it's reversed, which really was a surprise. And then for the next 4 years, we spent really looking at mechanism. And if you give a high-fat diet and then you give this siRNA, you can actually even reverse, and you can and you intercross these mice with a loss of spectrin in the liver. You can rescue this. So that's very complicated. You'll say none of this makes sense. And the heterozygous get cancer. How is it that you give siRNA and then knock it out in the liver and the cancers go away? And is this a mouse phenomenon? Does this happen in humans? And we were able to prove, we are able to do a lot of things. We're able to prove through single cell sequence of NASH HCCs that this is hepatocyte specific from UCLA data. And then take this to a human NASH in vitro study where you can culture NASH patients' livers in culture in microfluidic study done in Cambridge, England, where you culture these cells and they get NASH seen on the left. And then you give the siRNA, the cells are still viable, but the NASH is reversed, seen on the bottom right panel, and also the microarray. The left panel, SI control, shows a lot of smooth muscle actin, a lot of fatty liver disease, and the siRNA shows it disappearing. So you'll say, wow, siRNA knockout in the liver, is this safe? What's the mechanism? Why does this occur? And can we do this in humans? And is it going to be deleterious? And, you know, I think pre-COVID, I would have been very hesitant, but COVID is an RNA vaccine, and we've now come to RNA, the RNA field. So I have some thoughts, but what about this mechanism? And I think, Sanjay, you will be rather excited by this, because we found along the way, this is beautiful paper in Nature from 2020, which identified the liver, brain, gut, neural arc, maintaining a T regulatory cell niche through ALDH1 and TGF beta in the gut. So aldehyde dehydrogenase 1, alcohol dehydrogenase 1 is really controlled in these APC cells, and Tregs, TGF beta driven Tregs, really control these vagal efferents to really alter inflammatory bowel disease. And this is a communication system with the gut microbiome to the liver, and the organ with the largest innervation of the vagus is the liver in the body, and the liver, then the vagal afferents then go to the nucleus tractus solitarius, and then the efferents then communicate the signal. So you have this intricate system of gut microbiome, liver, brain axis. And this really dropped me into the bioelectronic medicine program and institute at Northwell and Feinstein, because they have an intricate knowledge of this whole vagal afferent and efferent system. And they can give sonography to various aspects of the body and stimulate and block different aspects of the vagus nerve, which is sort of really exciting for us, because could you modulate obesity through that? But what about this gut microbiome? So these are some of the bacteria that are involved on a high-fat diet with a liver-specific knockout. Remember that this is the one that does not develop NASH. We found Anaerotignum propionicum, I don't know how to pronounce these, Sanjay, you probably know, is really high compared to the high-fat controls, which are really fat, right? And the heterozygous, which are really fat and get the cancers. And then strep pseudoporcinus is completely lost when you lose the obesity. And then this ruminus is again erased. So these are interesting bacteria and microbiome, and we are pursuing this very actively. So what I've shown you is that these spectrin aldehyde mutants develop NASH and HCC, and that alcohol induces fibrosis. I haven't shown you that. The mechanism is through SRABP1 activity. It's a long mechanism, which I haven't gone into. But suffice it to say that this siRNA rescues this. Why is this exciting? Well, I don't know if you're familiar with the New England Journal paper and the phase 3 and the FDA approval of porphyria treatment through siRNAs through that is a Swedish company, and it's a large consortium here. And Monty Bissell, who visited me many years ago, said, Lopa, you're right in that space with this siRNA, and you should take it further. We've had this New England Journal paper. It's really there. And I still was very reluctant because I wondered if this was safe. Well, in the neural system, siRNAs to spectrin have been tested in 2 other systems, and there's a long history of them, and they're safe. So you disturb a well-known protein, it's like an actin, and it's present throughout the body, and yet nothing happens when you disturb it, but only good things, and pathways then reorganize themselves, and so it really rescues this. And I can give you reasons why we think this happens and why it's so safe. And it's not just us who believe it's safe, but in the in the CNS and neurogenetics, and they believe for motor neuron disease, it could be very safe. And so there's this SRABP pathway which is disrupted, and that is and that these aldehydes, which really bind to these spectrin fragments that are in the heterozygous state, divert away from SMAD3, and they then generate this pro-fibrotic, pro-tumorigenic events in tissue beta signaling. So we think it's a convergence of three pathways: TNF alpha, alcohol, and tissue beta under pathological conditions. This is all abnormal that generates this escape from a negative, normal negative feedback loop which exists in you and I to prevent lipid accumulation, prevent liver injury, NASH, and hepatocellular cancer. And the question is whether we can reverse this. So Bob Miller actually has taken this to a number of pharmaceuticals, and they asked us, they said, well, you know, the spectrum is everywhere and we don't know whether it's safe. And we said, well, the knockouts in the mouse studies show that if you have the genetics, it's very different. But if you have an siRNA, it's like a mop. And this siRNA behaves a bit like the COVID vaccine. We never expected an RNA vaccine to behave in this way. RNA behaves in a very specific way, and we think it behaves like a mop, preventing these fragments binding to toxic compounds, and really mops up the process. And we think that's where we are. And we think we've gone from discovery to perhaps getting towards the right panel of tissue blood markers, perhaps towards the right patient to get to this area seen on the right, to cures, for which we now have that for stage 4 melanoma and many other cancers which were considered incurable even as recently as 5 years ago. And, you know, we've had a president of the United States prove that for many cancers, others have proven, but for hepatocellular cancer, we're still here in this elusive space. So this is a great opportunity for all of us. This is this happened with an army of people: Kitty Shetty at Maryland, Linda Wong, who's a surgeon at Hawaii, Shulin Lee at MD Anderson, Herbert Yu, who has a huge cohort of patients, blood and plasma, which he looks at for SNPs, companies like Aitoria, the UCLA, SomaLogic, and University of Cambridge. And then, of course, a lot of people at George Washington University, and now at Northwell. And it's been great to have Sanjay join, and David Bernstein join, and Ben Dar join this program, as Jim Crawford has as well. And of course, this is funded by a number of NCI grants, thank goodness, and also a lot of startup money. When I had the program project, which NCI recently approached us about renewing, one of my neighbors had us have a dinner with Justice Ginsburg. And so we had a dinner at the Cosmos Club that I belong to for the whole program project advisory board. And this is the instrumental lab, Shuyun Rao and Wilma Jaganuri, who's been with me for over 20 years, who has been instrumental, and of course, Justice Ginsburg and Nina, my neighbor, over this wonderful dinner we had in, I think, 2016 for this. So thank you all for listening to this.